Yes, good evening to everyone. So, welcome to the day two session on target modeling and validation in online internship and certificate course on computer aided drug and vaccine design. So, before going to today's session, can we recap the last session topics? So, what exactly did we discuss in the last session? So, can we interact through polling? Okay, so I'm going to raise a poll now. So I just raised you a first question. Just answer for that. The first question is, which is 1D format of chemical compound? So for this, four options are provided. So complete your polling within 30 seconds. Which is 1D format of chemical compound? Yes, in fact, this is most important. I initially asked you a descriptive type, like whatever you know. Now I am giving you choices. It's easy to answer. I have taken you to the NCBA PubChem and I have downloaded few formats. You remember? So, what is the 1D structure of chemical compound? See, at least two participants have interacted. The others, if you don't answer, you get yes. Still 10 more seconds left out. Just interact, inky pong if you don't know. So, you should have watched the videos for answering the correct choice. So, just 3 seconds left out. Okay, I'm going to end the poll. So the majority have voted Smiles format. Smiles, the small molecule input line entry system. The expansion of Smiles is small molecule input lines entry system. This is the correct answer. Okay, the Smiles is the 1D format. Then how about all these three? These are either 2D formats or 3D formats of chemical compound. Okay, now let me go ahead with the launching the second poll. So the second question is, which is a protein-protein interaction database? PPI, simply we abbreviate as PPI. For this, I gave 20 seconds. Just answer. Last time only 9 votes we got. This time, at least increase the number of votes. Come fast, just 10 seconds. 5 seconds. Going to enter poll. See, only two additional votes. Okay, not bad. So 13 votes. But you see, nearly 30 participants are here, but only 13 are answering for this. So I don't know why you're showing your involvement. Try to interact. That's most important. Right or wrong, you can correct in future. Okay, the majority of them have voted for string. You remember? I showed you a kind of a database. I told you this is a protein-protein interaction database. Initially, I searched with an example like trap1. I told you trap1 is nothing but heat shock protein 75. After showing that molecule, initially you can see that trap1 is interacting with 10 protein molecules. And then I've gone to the settings, I increased the number to 100. Then we got 100 proteins interacting with trap1 protein. If I've attended last session, you may recall all this. The answer is string. How about the wrong answers? The NCBF WebChem is for chemical compound database. That is not for protein-protein interaction. RCSB PDB is a protein structure database. NCBA is again a protein structure database. Okay, so good, good interaction from the 13 participants. Even I anticipate other two interact well. Okay, even can answer up to the descriptive type. Actually, last time we have missed a few databases, isn't it? Can we look through the databases? Then we can start the session two. Okay, the next thing what we can able to see is the UniProt. Okay, so the next thing today we'll be discussing about UniProt. And each and every 2 days, I'll be circulating you the feedback link. Kindly answer to the feedback link. And I'll be sharing you the recorded videos on each and every day within 12 hours. Within 12 hours, I'll be sharing you. Because the recorded video has to be processed and it has to store in the drive. Okay, so it needs a minimum of 12 hours. Now, let me go ahead with the UniProt. So the UniProt is a database from UK. Okay, and this UniProt resource, you find plenty of databases like UniProt KB, okay, and UniRef, UniProtein Archive. You know, this is actually a fully annotation protein sequence database. So I was explaining you something on trap1 protein, okay, and I told you the trap1 is heat shock protein 75. Okay, if you want to know more about this protein, okay, like how you want to know more information about a chemical compound, I told you to search in NCBA PubChem. Okay, if you go to the NCBA PubChem, you can able to get full details like physical chemical properties, and you get spectral information, you get the vendors information, and the you'll be getting much more information. Okay, for chemical compounds, drugs, and even phytochemical compounds available. Okay, and similarly, this Uniprot you can find protein information. Yeah, and participants, so kindly mute, okay? Unless if you have it doubts, you just type the message in the chat box. I can address you whenever I'm coming on, okay? And accept our invitation in the Google Meet. If you accept, if you click yes, so each and every time there is no need to admit, okay? I believe I admitted today, Kalaivani. So try to accept our Google invitation and join in the WhatsApp group so that I can send you relevant kind of information, okay? Whatever I can't able to post it now. And now let me go ahead with this Uniprot. I was explaining something about Uniprot, and the most important is practice. I'm just training you for 1 hour, but you have to practice for 2 to 3 hours from the video, whatever I'm going to send, and whatever links I'm sending to you. If you have any doubts, I give you a chance or okay to present your screen and clarify the doubts. For example, if I provide you demo to download and install a software, but you don't know how to make it, I can also ask you to share your screen, and you can any kind of troubleshooting I can make it. Okay, so but everything you should inform me in advance because we are packed with so many concepts. So now let me go ahead with the trap one protein. Whatever I searched in the universal protein knowledge base, that is the expansion, universal protein knowledge base. Here you get full detail annotation. For example, now I want to go to this Homo sapiens, human kind of a protein, where this size is around 704 amino acids. So for this, we will be getting inside this Uniprot ID entry. The Uniprot ID entry is around 6 letters. This is actually an alphanumeric ID. Like, however, you find the PDB ID is around 4 letter ID. Okay, this is like a 6 letter ID. And in this 6 letter ID, you can able to see if you either you have to type the 6 letter ID or you have to type the protein name and species name. Okay, so these are all considered as keywords or database identifiers. So here you can able to see the function, names and taxonomy, subcellular location, disease and variants, post-translational modification, expression, interaction, structure. Okay, so all these things. For example, if I want to get protein-protein interaction, okay, you have to simply click this interaction. Here you can see the binary interactions. And now you can see the protein-protein interactions. Okay, like biograde, CORM, database of interacting proteins, fun group, intact, mint string. Okay, so these are the PPI databases. And similarly, if you go to the structure, you can get the 3D structure of the proteins. If you want to download the 3D structure of proteins, okay, so we can able to download based on our choice. For example, if you see here, we have plenty of PDB IDs. So these are the 3D structures actually posted from X-ray technique. And if you see which is the best you can able to use, first you have to check the amino acids. If you see the amino acids of ranging from 60 to 561, and the entire total size, it comes around 704, I believe. So if you want to download the entire thing, this is the one. Okay, but this is a predicted one. AlphaFold DB. This is an artificial intelligence enabled database. Actually, through the AI, the 3D structure is predicted and is posted. So only we are getting like AlphaFold DB. Okay, only here you can able to see the structure is predicted from 1 to 704. The total amino acids are used for prediction. You got it? So this is something about what to choose and what not to choose. For example, if your binding sites are lying in this range, 60 to 704, okay, then you can choose the protein structure to be downloaded from the resolution. If you see the resolution, the lowest resolution is the best magnification, best quality protein. So in this case, if you see 1.5 angstrom is the lowest resolution. And if your binding sites are lying in this range, we can use this. Or else you can check in the 60 to 704 and check the which is the least resolution. And you can download the structure and you can use it for docking, simulation, and other purposes. Okay. So this is a fully annotated protein sequence, and you can find structures and protein-protein interaction. So much more information you can find from this database like Uniprot, you know. And one more thing, I like to inform that the majority of the participants are second year and third year. Okay, so I am taking from the basics. But if you want me to take something advanced, you have to provide the feedback manually to my WhatsApp number like that. Okay, so now let me go ahead with the next thing. So now I'm going to the next database is we already seen stitch database, string database. Okay, so this is actually Keck database. I told you this is now made commercial. Okay, so because of the source is from Japan. Okay, now we can see something called target database. Okay, so this is actually therapeutic target database. And yesterday, you remember I have shown you drug bank. That is one more chemical compound database. Okay, and this is therapeutic target database. It is taking some time to load. So meanwhile, I can show you one more chemical compound database, Zinc 20. You know, the Zinc 20 is the largest freely publishable database where you can see 1 billion commercially available compounds. This is the largest database of compounds. Okay, yeah, so let me skip. I'm getting this. Find all animal icons. I have to choose animal. Okay, blue. It's most challenging still. Let me come across what else they want. Drag one character to the matching character. Okay, this is Chick 20 database. Here you can able to find something called branches. If you click branches, it take you to a kind of a library of molecules. Whatever we have 1 billion compounds. Okay, it is mapped in this table. In this table, you can able to see the compounds are mapped with respect to two physical properties of compounds. For example, here if you see in the x-axis, the compounds are mapped from molecular weight 200. to 500 and above 500. But whereas if you see in y-axis, the log partition coefficient scale is from -1 to 5 and above 5. For example, if I want to download the compounds of molecular weight 350 and partition coefficient 2.5, then we have to map accordingly. Here, if you select this, this is 39 million compounds. 39 million compounds. So if you want to download this all the 39 million compounds, that is which is mapping the molecular weight 350 and partition coefficient 2.5, you have to simply click download. This is the option to download. Now all the molecules are downloaded in a single data set. Got it? So all the molecules are going to download in the form of a single file. So this is one such chemical compound database. Okay, if you have a good workstation, high capacity server, then it can use these compounds for docking or any other purposes. Okay, so now let me go ahead with the next thing. Like today we'll be discussing on the target modeling and validation. So first, something about the research. So here, for the PhD scholars, they receive a stipend of around 18,000, and if they publish any research paper, they'll be getting 20,000 as a research stipend. And the MTech enrollment is going on. Out of 18 seats, 16 seats are filled. Only we have two more seats. The advantages are like you can see there is a reduction of around 30% tuition fee for eligible candidates. And we provide industry skill training. And 6 out of 18 students get merit scholarship. Out of remaining 10 students, one get merit coming scholarship. So we sponsor for chemicals, agents, etc. Okay. So this is something about the MTech program in SVC. Only two seats left out of total 18 seats. Now let me go ahead with this context. So if you see today we'll be discussing on protein structure prediction, and we discuss the levels of protein structure prediction. Then we come across the computational method like homology modeling. This is first method. Then we discuss on threading, the second method. The third method is ab initio modeling. And we also discuss how to validate the modeled protein structures. If we don't have any 3D structure in database, then we go use the computational methods, computational tools for modeling. Okay, so after modeling these 3D structures in the form of PDB file, we have to validate whether the quality of a protein, whatever predicted, is accurate or not. Okay, now let me go ahead with this protein structure prediction. You know, these are the levels of protein structure prediction. Primary. So this is the first level of protein structure prediction, where you can able to see the protein structure is present in the form of amino acid sequence. Totally we have 20 amino acids. For each and every 20 amino acids, okay, each and every amino acid is represented by a single letter. For example, whatever we represent as A is alanine. F stands for phenylalanine. D stands for aspartic acid. V stands for valine. Like this, so can study all this information from biochemistry textbook. Okay, what are the 20 amino acids and what are the single letters? Now let me come across the secondary structure. In secondary structure, we have alpha helices, beta sheets. The alpha helices contribute more than 50% of secondary structures. If you see the alpha helices, we have subtypes like alpha 310, okay, alpha 310, 5 alpha helices, okay, and general alpha helices we like this we have, okay, and even in alpha helices we have left-handed alpha helices, right-handed alpha helices. These are the subtypes. Again, I'm telling you, 310 alpha helices, 5 helices, left-handed alpha helices, right-handed alpha helices. Similarly, in beta sheets we have parallel and anti-parallel. So these are the types of beta sheets. Other than these structures, we have loops, coils, turns, etc. So these are the types of secondary structures in protein. Like if you know the primary structure, it one can able to predict the secondary structure. And immediately we want to find this tertiary. In between tertiary, we have one more structure. You know the name of structure? The structure in between secondary and tertiary is what we call it as super secondary. Is called super secondary. In super secondary structures, okay, we find protein structures in the form of motifs. So in motifs, we have beta, alpha, beta motif. This is one example. Like this, we have Greeky motif and we have jelly roll motif. Like this, we have types of examples in motif. Motif is nothing but a combination of secondary structures. For example, in this case, after beta, if you find alpha and again beta, this is now actually is called beta alpha beta motif. And whatever we can find this is an alpha helix. The alpha helix is generally represented by this, just because it's coming like this. Okay, so this is actually what is this? So this is actually right-handed. So this is left-handed alpha helix. Okay, and like this, if you see the beta shades, the topology diagram looks like this. It looks like an arrow mark. Okay, and generally you find N terminal here, C terminal here. Similarly here also you can find N terminal, and here you can find the C terminal. So these we call it as beta shades, and this we call it as alpha helix. So you find beta followed by alpha is connected, and this alpha is further connected to one more beta. Okay, so this is what beta alpha beta mode. The next thing is the tertiary motif. The tertiary structure. In the tertiary structure, we can able to find a protein structure in the form of chain. We have only one chain in tertiary structure. Of course, the 3D structure of protein starts from tertiary. This is also tertiary, but this we call it as quaternary, where here we can find more than one chains. Generally, in this drug design, okay, in the protein structures, generally we call this one particular thing as a chain or domain. So here in quaternary, we have multiple chains or multiple domains. So for example, so this particular color belongs to domain 1, and this is domain 2, and without shade is domain 3, and this is domain 4. Totally we have 4 domains connected together. If you have more than one domain, we call it as quaternary. Got it? So totally, if you see the levels of protein structure, we have totally 5 levels of protein structure. Now let me discuss something on amino acids in protein. I told you we have 20 amino acids. In any biochemistry textbooks, you can find a single letter for the amino acids. For example, R for arginine. Like this, you can see examples, and you can also see how these are classified. Now let me go to the next slide about peptide bond. What is peptide bond? How is it formed? If two amino acids, okay, if two amino acids are linked with each other, then a water molecule is going to be eliminated. See this yellow part? Here, this OH is interacting with the H of another amino acid. So you can see a release of water molecule. If water molecule is released, then the C double bond O is going to bond with NH. You can see this bond. This bond is what we call it as peptide bond. Got it? Now let me go ahead with this secondary structure of peptide. So if you see, if a peptide bond is formed, okay, between the C double bond O and NH, so whatever I show you like Ca is nothing but carbon alpha. Ca is a carbon alpha. So now let me discuss something like chimeric carbon atom is generally, we call it as carbon alpha. Here if you see this kind of a peptide bond, okay, the angle between NH and C alpha is what we call it as pi angle. Pi angle. And the bond between C alpha and C double bond O, we call phi is, this is psi actually. This is psi. Let me take an eraser and so this is actually psi. So take it when yeah. So phi is actually we represent like this, isn't it? This is a phi angle, and this is a psi angle. And omega is nothing but the bond between C double bond O and NH. Now we can see the secondary structures, the type of secondary structures. The alpha helix, okay, a form of a default alpha helix, has a definite phi, psi, and omega angles. Similarly, you can able to find so whatever we call alpha helix is defaultly a right-handed alpha helix. Okay, and this is specifically left-handed alpha helix. And 310 helix, we have it here at a variation with respect to phi psi. So similarly, you can find the angles vary with respect to other secondary structures. Now let me give you an explanation of secondary structure of alpha helix. How the secondary structure is predicted from primary structure. If you see, I told you the most occurring secondary structure is alpha helix. No doubt, more than 51% of proteins uh, have this alpha helix. If you see this, okay, in the alpha helix, okay, the most favoring amino acids are methionine, alanine, arginine, lysine, and leucine. So generally, for forming an alpha helix, we need around 8 to 15 amino acids. This short alpha helix can also be of 5 amino acid in length. For example, if you have a small alpha helix and if you have this in the sequence, whatever I showed you, all these amino acids in the sequence, like if this is coming under position 1, amino acid position 1, this is amino acid position 2, 3, 4, 5, 6. Okay, all these 6 amino acids lying in this most favorable amino acids which can form the alpha helix. Okay, so definitely this sequence can form the alpha helix. But there are some unfavorable amino acids. Like what if you find these amino acids in the sequence, this won't provide you the alpha helix structure. Instead, it can provide beta sheet or loop tones, anything like that. The amino acids which are not most favored to form alpha helix is proline and glycine. Proline and glycine. So after this amino acids, you can also follow these amino acids also follow the similar patterns. Okay, so for predicting the secondary structure from primary structure, the sequence of amino acids determines to choose whether it's going to form alpha helix, beta sheets, or loop stones or coils. Now let me explain you something about the structure with an example. If you discuss an example like globin family of proteins, you know, in this example, whatever I've shown is a hemoglobin A chain, hemoglobin B chain, myoglobin of swine. These two or belongs to human, the first two. The third belongs to the swine. If you see, the hemoglobin is different from the myoglobin, isn't it? And that too, if you take a different species, definitely it varies. But according to the number of amino acids matching with each other, if you compare with each other, if you see, hemoglobin of A chain is more resembling to the myoglobin of swine. So these are the wonders, if you take all these protein structures, the protein structure looks very similar. You can't distinguish this blood protein hemoglobin with muscle protein myoglobin. The structure wise you find same, but only the sequence alone varies. Similarly, if you compare with the leguminous hemoglobin, leguminous, chickpea plant, it is a plant. The plant, the protein has same structure of human blood protein. But if you compare these sequences, the amino acid sequence, the sequence vary with even up to 16% similarity. So like leguminous hemoglobin, bacillus hemoglobin, sea urchin hemoglobin. Okay, so it can be from any species. All the species share the common structure. The structure is very similar. You can't distinguish whether it is a plant or it's a bacteria or is it from human, but the sequence wise you can find variation. So this is how you have to understand. Okay, the sequence is different, structure is different. In a family of proteins, the structure usually remains same. Now we discuss like how the structure is discovered from the sample. If you want to use X-ray crystallography, so we have to take a sample of protein and we have to crystallize. Unless we get a crystal, we can't able to get a 3D structure. So this is most difficult task. The protein is a macromolecule protein, a macromolecular biomolecule. So getting a crystal is not an easy task. Crystallization process nothing but mostly of evaporation to heat, and the magma is going to create a kind of a crystals in the magma. Remember, or during the saturation, super saturation, these are the concepts we study. So if only if you form a crystal, then we can pass the X-rays. If you pass the X-rays, we get only this diffraction pattern. Like if we take an X-ray, how we find only the bones are visible, the bones are shown in white color. The remaining things show the flesh or all the things shown in gray color or in black color, isn't it? Similarly, even the protein crystal, if we do the diffraction pattern, we get only black dots in the white space. Each and every black dot can be an amino acid also, but you don't know which amino acid is followed by another amino acid. Okay, in the protein sequence. We need, of course, the 3D structure, not just the sequence of the protein. If you need a sequence of a protein, use sequences. Protein sequencer for sequencing. If you need a 3D structure, of course, we have to go with only these kind of techniques: X-ray crystallography, NMR, cryo-electron microscopy. Now, let me explain at least one technique clearly how we get a 3D structure with X-ray crystallography. So, one such patterns we get, okay, this is an intermediate kind of files we generate, and later we try to get electron density maps. From the diffraction patterns, we can able to generate electron density maps, and finally during simulation, this is the process actually we make to convert the electron density maps into atomic model. Generally, the atomic models are present in PDB format. Any file name .pdb is a 3D structure of the protein. Okay, so this is how the 3D structure is predicted from the protein crystal source. Now let me explain you the methods of protein structure prediction. I told you the 3D structure of prediction starts from tertiary. Why it is start from tertiary? Because even a single chain, some proteins have a single chain. If you have a single chain or single domain, still we call it as a 3D structure. Okay, but few proteins have multiple chains. Then even it comes under 3D structure. Now let me come across the most important methods for computational methods for protein structure prediction: homology modeling, threading, and ab initio. The most widely used online tool to predict protein and homology modeling is Swiss model. I am going to provide a demo on that, and the threading using PHYRE. And the ab initio, you have plenty of tools. Many try to use Anolia, Rosetta, okay, Procheck. So what if these are all the popular tools? Now let me explain about steps in homology modeling. The steps in homology modeling you can find from any web resources. But let me teach you like how to practice what else we can able to know. So the homology modeling can be done in freewares as well as in commercial softwares. This is a window from Discovery Studio Modeler. So we have plenty of tools to model the structure using homology modeling. This is a Discovery Studio Modeler tool, where if you simply paste the sequence, this is the sequence actually nearly 185 amino acids. Just like that, if you paste it and if you click routine structure prediction, obviously it predicts the structure. So you get these kind of a results, and you can finally can download a protein structure. Okay, this is from Discovery Studio Modeler. So the final structure will be like this. So for whatever structure we predict from the modeler will be represented in form of dope score. Based on dope score, one can able to check the quality of the protein. Structure validation has to be done through Ramachandran plot. So more about this we'll be discussing in subsequent slides. So this is what I'm going to train you. That is practicing the homology modeling using Swiss modeler. So Swiss modeler is a freeware tool. Okay, so initially one has to click the start modeling, and then you have to paste the sequence. We paste an amino acid sequence for the sequence we don't have any 3D structures in any databases. Then this software tool is going to predict the structure for us. Okay, so while predicting a structure, we also have one intermediate step. It asks you to select one template. For example, the fat persons. Okay, one who is more obese. If we go to a branded shops, if you want to procure Van Heusen brand, Allen Solly, whether we can able to find the brands for them? No, it's not possible, isn't it? So what we that obvious people can do, they can procure bit piece, and then whatever earlier template they have, the stitch cloth or even some kind of ready-made, which is more suitable for them, they give that. So that is a kind of a template, and whatever bit piece they have, that is a kind of a target sequence for which sequence we want to predict a structure. So that is we provide the sequence alone as an input, and then we predict through predicting a template. Okay, so I show you step by step. Now, can I take you directly to the window, browser window? Am I audible to everyone? Yes or no? Kindly interact. Yes? No?
Yes, sir.
Yes, sir.
Good, thank you. Because I should know whether I am in internet or not. Okay. Now let me take you to the first thing is Swiss modeler. The Swiss modeler is simply type Google in Swiss modeler. It takes us to swissmodel.expasit.org. This is the URL. Okay, so we got this kind of window start modeling. Simply click this start modeling. Now we have to paste one protein sequence. Whatever protein sequence you're going to paste, that's sequence. This trap one, for the trap one, uh, we don't have a structure for 1 to 704 amino acid. We have one structure, but that is predicted from alpha fold DB. But if I want to predict a structure, no, actually, even at least we have these PDB structures, isn't it? This is from human. So for human, we have at least few structures. If you take trap one of mice, you know, for the trap one of mice, I'm directly taking you inside. We don't have any PDB structures. See, if I click the structure, I don't find a PDB ID. Instead, I find only the alpha fold DB. At least there we find some PDB IDs. So where the structures are deposited from the gold standard techniques like X-ray crystallography and ML. But here we don't have. It's just a computational predicted method. Why can't we predict for these kind of protein sequence? So for predicting the structure from the sequence, we have a sequence here. Click this sequence. Now we find this is 706 amino acid. So if you want to download this amino acid sequence in FASTA format, click this download. So now we got this sequence. Either copy this amino acid sequence alone, or even if you copy the entire FASTA format, no problem at all. For Swiss model, you simply have to paste the entire FASTA. Even if you paste FASTA, it takes only the amino acid sequence. Okay, and you can give some project name. The project name is actually, I am going to predict for trap on mouse. for the structure which is not present in Uniprot. And we can give an optional mail. Like this kind of a prediction, a protein structure prediction can take 5 minutes, or it may take beyond that based on the amino acid sequence of protein. If you have a small sequence protein, the prediction is instant. But if you provide a large size protein, the 706 is large, 100 amino acid is small. Okay, so as I'm giving a large amino acid protein, it takes some time obviously. So better give a mail where this gives the results are generated and it will be sent to your email ID. So you can click the hyperlink to see the results. Now we have two options. One is search for templates. The search for templates is an intermediate step. The system can also predict model from the best template. For example, when I told you if a person want to procure Van Heusen uh, pant, the Van Heusen, whatever he stitched some 6 months before, that might be a more fitting when compared to a pant which he has stitched 5 years before. So we have to select a suitable template for the bit piece to be stitched, isn't it? So if you want to select any template, you can use this intermediate step. Or if you trust the server which can able to choose a best template, you simply click the build model. If you click build model, for the sequence for which the structure is unknown, the structure will be predicted from this. So meanwhile, we can go to the next tool. It takes some time, I told you now, so at least 5 minutes. So I'm going to take you to the next tool called for threading. We see something. What is that? Uh, for threading, we'll be trying to use iTaser. Okay, iTaser. iTaser server for uses threading concept. iTaser refers to iterative threading assembly refinement. So for this, it is a must to register. Okay, it is must to register, and after registering, you have to log in using username and password. And participant, if I am disconnected because of internet frequency, you can give a missed call so that I will be getting alerted. Okay, even this is one of the option you can choose to connect continuously. Okay, now let me come across. I've already registered, so I have a user ID and password for this. Okay, and now you have to simply paste the sequence. The sequence is this, or you have to upload the thing. Here, according to that, they're asking to paste in FASTA format. So whatever you pasted is in FASTA format, no doubt. So if you want to give anything like that, nice. But the disadvantage of this is that more than it takes time from 1 day to 7 days based on the length of amino acid sequence. For the Swiss model, it takes just 5 minutes, but for here, it takes 1 to 7 days. So you must provide email and password. This is the reason why they have made it as mandatory. And simply have to click the sign ITS. Okay. So whatever we receive after 1 week or within 1 week, even the results will be sent to our email ID where we can open the hyperlink and can see the result. Now still it's running the Swiss model. Okay, if we get the results, I can show you. So meanwhile, I can tell you like what else we can able to see from here. So threading. Then what is the difference between the threading and ab initio? I just explain you with simple terms. In homology modeling, we take one target sequence and one template PDB, and we predict the structure from the template PDB. One target sequence, one template PDB. This is sufficient to download one model. We get two models, we can check which is the best model and we can use this model for docking, MD simulation, etc. So these are the other methods of computer truck design. Now let me come across threading. Instead of predicting one template, here you will be getting multiple templates. Use one target sequence, no doubt, but here you use multiple templates. Because of multiple templates, the prediction is more precise when compared to homology model. Okay, so these are the advantages and disadvantages. The disadvantages, it takes more time to generate the results, and it generates multiple models. Here we get 22 models and we can select which is the best. But here we get around 10 models. You can select based on our choice. Okay, this is something about difference between homology modeling and threading. This is the window I showed you earlier, and this is the pipeline. Okay, so we can come to the next method, that is ab initio prediction method. In ab initio, unlike homology modeling and threading, we predict the protein structure in phase-wise manner. So if you see the threading, even it is predicting from only primary amino acid sequence, but it predicts automatically to a final tertiary or quaternary structure. But here in ab initio, we have to initiate from the sequence, primary structure, and each and every level you have to do some steps. So this is something about ab initio. I forgot to show you something about the output page of the Fiery. In Fiery, you'll be getting the outputs like this, many rough models. You can download the models. Now about ab initio. In ab initio, actually we are predicting from the atom-wise approach. For example, so the proteins or amino acids are bonded together in protein through bonded interactions and non-bonded interactions. The bonded interactions are generally through bond lengths, bond angles, torsion angles. More about this we'll be discussing in subsequent sessions. Like bonded interactions, even non-bonded interactions also plays an important role to determine the protein structure. Okay, so this is something about these models. Now we are getting this output. Meanwhile, one participant has raised the doubt. No, if you have any doubts, you can ask me. I got some kind of interaction from your side. No, then I can
S-
continue. Yeah, yeah, you can,
Sir,
you can. Yeah, I can.
sir, I have a doubt.
Speak something if you have doubt.
Uh, what is the meaning of ab initio, sir?
What is
Ab initio.
uh, what is happening? That's what I told you, isn't it? We develop the structure from stepwise. Okay, so we develop the structure from the stepwise. We have several tools for ab initio structure prediction. Okay, like what if, what check, okay, anolia. So we have different tools to predict ab initio, and we predict structures by phase wise. Like we have seen the levels of protein structures. So it built from the atom wise. Okay, and we predict the final structure. Now let me go ahead. Anything else?
No, sir.
Yeah, you are asking about theoretical things. I just explained up to this much. You can find plenty of information, plenty of internet sources. Now let me go ahead with these models. So if you see these models, so if you see the models, we generated two models, model one and model two. Okay. So if you see the which is the best model, we can able to select the best model. No, if you have some other kind of an opinion, you can ask me. The participant, you have some other kind of an answers. You want to explain something about ab initio, you can explain. You have any kind of contradictions with whatever I spoke earlier? Do you have any contradictions? Yes or no?
No, sir.
Okay, then let me continue. Okay, so if you are speaking with respect to this, how to predict the results, okay, from homology modeling is in Swiss model, I told you simply paste the sequence, get a structure by clicking a button called build model. Here we got two models. The one model, what we see here is the sequence identity is 78.53 percentage. The next model is 88.49 percentage. So if you see, if you ask me which is the best model to be used, then I can tell you the model 2 is best because the coverage is 88.49 with respect to template PDB. You got it. So if you find major coverage with the template PDB, then you can download this. To download, simply click this and download PDB format. Now, if I click this PDB, okay, it asks you to download somewhere. We already have one folder. Now I'm going to save like model underscore 02 PDB. And here in this tool, in this online tools, we can also find structure assessment. This, if you click this structure assessment, it directly take you to Ramachandran plot. I'm just going to click this structure assessment. You heard this name, Ramachandran? Anyone heard about this name, Ramachandran? Who is Ramachandran? G.N. Ramachandran.
He started the Green Revolution.
Ah, he started the
Green Revolution.
Green Revolution. Good. So now let me
Uh,
explain more about him. So he is the father of bioinformatics in India. So this is also we can also describe him like that. You know what exactly he has done. So Green Revolution, I think you are mentioning something about Swaminathan. Ah, okay, so that is something different here. So this is actually Ramachandran. The Ramachandran is a father of bioinformatics in India. Okay, if you see this kind of a plot, this is what we call this a Ramachandran plot. The Ramachandran plot is between 5 SSI. You remember NC alpha? I showed you three angles. I showed you phi and psi. So here, if you see this axis, okay, in this y-axis, you find 1 minus 182 plus 180. Similarly, minus 182 plus 180. Here we find four quadrants. This is left top quadrant, right top quadrant, and whatever you find here is the other two quadrants, bottom left and bottom right. So here you can find the dark green regions, and you can also find light shaded green region. You find few dots. What dot If you place it here, you can find this is lies in 31st position. If you place here, you can find the amino acid is 309, 39 position. So what do you understand? The 704 amino acids are now lying in this plot, in this Ramachandran plot. And if the amino acids are not coming or not covering in dark green or light green, and it is coming something out, this is unfavorable amino acids. These unfavorable amino acids are not going to contribute any of the secondary structures. You know, according to Ramachandran plot, I show you the Ramachandran plot and explain you. So this is how we had earlier we had like this, this is from project tool. The project tool is of course a part of ab initio prediction. So if you see this kind of a result, okay, we have most favored region shown is 91.7 percentage. So this is above 80 or 90 percentage. Just because it is above 90 percentage, whatever protein structure we have predicted is of high good quality. If it is of 80 percentage and above, it is good quality. So the good quality or high good quality can be used for further molecular docking or MD simulation, etc. Like if you want to go with the other competitive drug design methods, the protein structure quality should be good. If it is below 50 percentage, don't use that protein for any analysis. Just keep. Okay. Now, this is how we infer the quality of a protein. The quality, whatever inferring, is the stereochemical quality of a protein. Now, let me go to this slide. So I told you we have a dark green region and light green region. Here I am showing you in the form of red color and yellow color. So whatever amino acids lying in this, okay, so whatever amino acids lying in this, okay, is going to contribute beta sheets. And whatever amino acids, okay, lying in this region contribute left-handed alpha helix. And similarly, you can find right-handed alpha helix, and also you can see 310 helices, 5 helices, okay, all these things are lying in this. If you find amino acids somewhere here, these are unfavorable residues. Similarly, you can see here anti-parallel, parallel beta sheets. So like this, all the secondary structures you can able to find in this alpha-helix. This is how you can able to decide like what amino acids contribute to what structures. Got it? So these are not going to not contributing any structure. So this is unfavorable. Now, let me take you to the browser window and explain you further. So now if you see here, we have whatever we predicted for trap mines, we got 95.25. So I told you above 90% is high quality protein. So this you can use. So the purpose of showing you is to for structure assessment through stereochemical quality of protein. Here you can find the glycine plot separately. You know the glycine and proline or bad formers of alpha helices. These are not going to contribute alpha helix. That also you can understand. Okay, and including pre-proline. So the general plot you can find all amino acids. So this is how we can assess the quality of a protein using Ramsden plot. Now let me take you what else we have. I told you that 310 helices lie in this region and other 5 alpha helices. The second method to assess the quality of protein is by analyzing root mean square deviation. For example, if we have a 3D structure predicted from X-ray, NMR, any of the technique, and whatever protein structure we are predicting from either from homology modeling, for example, if this is 1 is 2, 3 over 5, see the first amino acid has shortest. If it is having a wider distance, the RMSD is closer to 1. Okay, the RMSD generally range from 0 to 1. If you have smaller distance, it is close to 0. But if you have a larger distance, it is nearing to 1. This is how you can understand, and this is the formula, okay, for calculating of RMSD. That is distance of second atom square minus distance of first atom square divided by number of atoms. So the third method to predict the quality of protein is predicting through critical assessment structure prediction tool, CASP tool. Okay, so if you see how the prediction, the quality is predicted using the CASP tool. So actually this CASP 2000 is using this whole prediction by this scientist. So according to that, uh, already the fold predictions done by the scientist, the structures quality or assessed. Okay, this is how the structure quality or assessed from the previously determined prediction methods from the scientist. And we have other tools to assess this structure. So this is actually, if you go to this URL, we have plenty of tools to assess the structure. Verify 3D, who, even ProCheck, the ProCheck is also generating the Ramachandran plot. The ProCheck tool is also generating the Ramachandran plot to assess the quality. So these are the references, these are the tools one can able to check and can practice. ProSA, Protein Structure Analysis. This is one more website where we have to simply upload the protein structure, whatever we have modeled, and we can analyze. So if you click analyze, you can check the quality of the protein. That's it. So I have completed today's session on how to predict the protein structure and how to assess the quality of a protein. Okay. If you have any doubts, you can ask me. There is also one more server called expassy.org. Okay, so that if you go to the expassy.org, so this is from Swiss Institute of Bioinformatics. Okay, we have even tools to predict secondary structure from primary sequence. Not only that, we also have tools for protein-protein interactions. So if you want to check the lipid-protein interactions, we have Swiss lipids. We have separately for Swiss drug design. Okay, so you can see plenty of the tools available, which is linked with the protein structures. So more about these tools, we'll be discussing in the subsequent sessions. So one can also go to this kind of expressity.org to interact with these tools. Okay. Now, welcome to the questionnaires. You can ask me any doubts, whatever you want. Do you have any queries? If no queries, thank you very much for the patience and interaction. So next time, I anticipate much more interaction. And I'll be circulating the feedback link to the WhatsApp group, and also I'll circulate this hyperlinks, whatever we have gone through. So, and also recorded videos within 12 hours. Okay. So, if you have any other doubts, accumulate it, and even I can ask in the next session. Thank you. If you have doubts, you can stay back. Others can leave.
Sir,
Yeah, ask me.
sir, this is not the related to the content, sir. Actually, I'm not added in the WhatsApp group still. May I get the link through some mails?
Actually, I have sent this link to the registered email ID. You do one thing. You can send me
Okay,
your
sir.
registered email ID or any other email IDs, because registered email ID I remember I have sent. You
Okay,
simply
sir.
have to simply send me the email ID for which you want to add. Okay.
Okay, sir. Thank you,
Thank
sir.
you. Okay. See you then. Bye.